The overall goal of the following experiment is to investigate the neural correlates of observing social interactions by using virtual characters interacting in social contexts in conjunction with fMRI recording. This is achieved by creating whole-body animated characters that interact with each other as hosts and guests in a business-like setting, and inanimate characters that serve as control conditions. Next, brain imaging data are recorded while participants observe virtual social interactions, and then data are analysed in order to identify brain regions that are part of the so-called social cognition network. Typical brain regions sensitive to observing and assessing social interactions include the superior temporal sulcus, lateral and medial frontal cortex, and the amygdala.
Because it allows investigation of the neurocorrelates of social cognition, this method might contribute to understanding possible causes of deficits in social behavior, which are observed in such clinical conditions as social phobia and autism.
The main advantage of this technique over existing methods such as those involving facial stimuli is that by using whole-body animated characters, it's possible to incorporate manipulations that more closely resemble real-life social interactions, such as formal physical touch and handshake. This is something that's not possible with just facial stimuli.
The task consists of a series of 10-second animated videos of non-verbal guest-host interactions in a business setting. The subject views the guest being greeted by a host, which is the social interaction condition, or by a cardboard cutout of a host, which is the no-social.